Hello students. So today we are going to learn the second part of the topic, plant growth and mineral nutrition. In this particular part of the chapter, we are going to learn development, plasticity, growth hormone, that is auxins, gibberellins, cytokinin, ethylene and abscisic acid. The growth hormones are the important part of the topic. Here we will be learning the physiological effects and uses of the growth hormones. Further, we learn about the development. Now you can see the chart which I have put up over here about plant cycles. In this, you can see how the seed germinates, plant grows, plant flowers, produces fruit, releases the seeds, and at a later stage, plant dies. And as the cyclic process continues. Also, you can see the flow chart of development, figure 7.7, taken from the text, where right from the beginning, seed germination, followed by meristem, cell division, plasmatic growth, cell elongation, cell maturation, mature cell, senescence, and finally death takes place. Now, coming to the first point of development, it refers to the ordered or progressive changes in shape, form, and degree of complexity. That is, based on the type of plant, the shape, structure, form, all changes. It includes all the changes occurring in sequence from the germination of the seed up to the senescence or death during the life cycle of the plants. That's what we have seen in this chart, as well as a simple image to understand the concept. Thus, development includes growth, morphogenesis, maturation, senescence. So growth, as we know, the seed which germinates or all the living organisms which grow. Morphogenesis, that is the external form, the appearance. Maturation, then it matures into the type of species, and finally senescence or death takes place. Moving further, next, plasticity. First point, it is a capacity of being molded, formed or modeled. It is the ability of the plants to form different kinds of structures in response to different environmental or internal stimuli in various phases of life. Now, as we know, all the species are different. All the species have got different morphological structure. So accordingly, they get molded or genetically they take their shape, size accordingly. And based on the environmental condition or stimuli, the changes takes place in the various stages of life. Further, in many plants, juvenile stage and mature stage show different forms of leaves in the same plant. Example, heterophyly in cotton, coriander, larkspur, that is delphinium. Now, when you say juvenile stage, this is the initial stage of the plant. Now, in certain species, the initial stage itself is different, or the leaf, the morphological structure of the leaf is different. Once it matures into a fully grown plant with fruits, flowers, etc. So, both the stages, the leaves are different, as example given in cotton or coriander or larkspur. Now, also, given the images of these species, coriander, all of you know, cotton, larkspur. Further, the environmental heterophily is shown by Ranunculus flabelliasis buttercup. That is, this Ranunculus flabelliasis is a, or otherwise commonly called as buttercup, also shows heterophily. The intrinsic plasticity is found in coriander and cotton. Heterophily is exhibited in the same plant in different growth phases or under different environmental condition. So you can see figure 7.8 from the text. Heterophily, juvenile stage, how the leaf appears. In the adult stage, when it get matures, how the leaf shape, the morphology is. Terrestrial habitat, and in case of water habitat. So you can see how the structure changes from the juvenile to the mature stage. Here, the buttercup flower of Ranunculus flagellosis. Moving further, know the scientist. Now, when we learn about these species, knowing about the scientist is equally important. The auxin is the first hormone to be discovered in plants. So, as we know, the first known plant hormone is auxin. Discovery of auxins dates back to 19th century when Charles Darwin in 1886 was studying tropism in plants. When we say tropism, if I give a few examples, phototrophism, geotrophism, hydrotrophism, right? He exposed canary grass coleoptile to unilateral light. He concluded that a growth stimulus is developed in the coleoptile tip and transmitted downwards to the growth zone. This has caused bending of tip towards the light, that is phototrophism. The Danish plant physiologist, that is Boysen Jensen, in 1910 cut off the coleoptile and inserted thin plate of gelatin between the tip and the cut stem. He observed that coleoptile's tip still bends towards unilateral light. What does that mean? Even after cutting the tip, the coleoptile, the growth occurs towards the sunlight, that is showing phototrophism. So here the scientists Charles Darwin and Peter Boysen Jensen, the canary grass on which Charles Darwin had conducted the experiment. Few more experiments conducted by Pahl and Wendt. Pahl in 1919 cut off the tip of coleoptile and replaced it asymmetrically on the cut coleoptile stem. He observed that coleoptile tip bent away from the side, bearing tip even in the dark. Now, Pahl, who conducted the experiment, showed that even in the darkness, the tip of the coleoptile, they move towards the light. F.W. Wendt in 1928 successfully isolated natural auxin from Avena coleoptile tips. He cut off the tip and placed them on some agar blocks. Then after certain period of time placed the agar blocks asymmetrically on the cut coleoptile stem that caused bending. He demonstrated the presence of substance which could diffuse into agar blocks. Wendt named the substance as auxin. Now here both the images of Arpad Pal and F.W. Wendt, and as Wendt did the experiment of Avena coleoptile, what he showed that this bending which takes place towards the sunlight or the phototrophism which is seen is due to certain hormone, and which he demonstrated the experiment and named the substance as auxin. Moving further, growth hormones. The term hormone was coined first by Starling in 1906 in Animal Physiology. The internal factors that influence growth are called growth hormones or growth regulators, as they inhibit, promote or modify the growth. Now, as we came to know about the hormones, which are growth hormones or growth regulators, they actually inhibit, that is control, promote, that is influence, or modify the growth, that is helps in making the changes which is seen in the growth of the species. Growth promoters are auxins, gibberellins, and cytokinins. Growth inhibitors in plants are ethylene, abscisic acid. All phytohormones are growth hormones. Now, phytohormones, that is the plant hormones, whether it is auxin, gibberellins, cytokinins, which are the promoters, or ethylene, abscisic acid, which is the inhibitor, all are growth hormones. Further, according to Thimen and Pincus in 1948, plant hormones are organic substances produced naturally in higher plants, affecting growth or other physiological functions at a site remote from its place of production and active in very minute amount. So, as we now know about hormones, Seaman and Pincus, they did further experimentation showing that plant hormones are organic substances which are occurred naturally in higher plants. They affect the growth and the functioning of the plants. They may be produced at a particular site or a remote region in the plant, and they affect the overall production, and they may be produced in a very small quantity or a minute amount. Further, growth hormones are transported through phloem parenchyma. Now this was concluded by Phillips in 1971 that these hormones are transported through the phloem region of parenchyma. Here I have posted the images of the scientists which are mentioned in the text: Ernest Henry Starling, Donald Phillips, Kenneth Timan, Gregory Goodwin Pincus. Now, first, auxins. Auxin means to grow. F.W. Wendt in 1931 used this term first. Auxin was synthesized in growing tips or meristematic regions of plants from where it is transferred to other parts of the plant. So, as F.W. Wendt did experimentation, he concluded that this auxin was found at the apical region or the tips of the growing region or the meristematic region, and further they transported to the other parts of the plants. The most common and important natural auxin is indole 3 acetic acid, IAA, that is the natural auxin which was found. It is the first hormone to be discovered in plants and is primarily responsible for cell elongation, that is the growth or length of the cell. It shows polar transport, basipetal transport in stem. Now, synthetic auxins like IBA, that is indole butyric acid, NAA, naphthalene acetic acid, 2,4-D, 2,4-dichlorophenoliacetic acid, etc. are used. So, as we can see, the IAA, indole 3 acetic acid, the natural auxin and first known hormone. Further, the synthetic auxins which was produced later artificially, there is IBA, NAA and 2,4-D. So here I had added the image of auxin, how chemically in chemical structure you can see, and indole 3 acetic acid, IAA, the first natural auxin. Moving further, physiological effects and application of auxin. The primary effect is cell enlargement. In most of the higher plants, growing apical bud inhibits the growth of lateral buds. This is called as apical dominance. Now, as the term apical dominance, the apex region. As we know, auxin helps in the growth of cell enlargement, whether it is root or stem. But at times, the apical bud inhibits the growth of the lateral buds. That is termed as apical dominance. Auxin stimulates growth of stem and root. Auxin induces multiplication of cells, hence used in tissue culture experiments to produce callus. It stimulates formation of lateral and adventitious roots. Now, as we know, the auxin helps in the growth of stem and roots, right? Or stimulates. But along with that, it also helps in multiplication of cells. So because of this, it can be used to carry out various experimentation to produce callus. What is callus? The undivided mass of cells, which can be used or helps to stimulate formation of lateral and adventitious roots. Further, these are marketed as synthetic herbicides. What is herbicide? Sites to kill. Herb, that is to kill the herbicides, unwanted herbicides or weeds which grows along with the other plant. Example, 2,4-D, 2,4-dichlorophenoliacetic acid. It kills dicot weeds without affecting monocot crop plants. Now, what are these monocot crop plants? The best example being rice, wheat, jawar, bajra, all the staple crops which are consumed as food, right? But along with these crops, unwanted plants grow as well. that are dicot weeds. So remove these weeds without harming the monocot crop. 2,4-D is used as the synthetic herbicides. Further, the seedless fruit like orange, lemon, grapes, banana, etc. are produced by the application of auxin, that is induced parthenocarpy. Now parthenocarpy, as we know, that is to produce fruits without undergoing fertilization. So by using or application of auxin, we can produce seedless fruit like orange, lemon, grapes, banana. Further, auxins promote cell division in cambium and also cause early differentiation of xylem and phloem. So here I added an image showing the growth of the coleoptile tip as we learned the experimentation early. So when sunlight is overhead, the IAA molecules produced by the apical meristem are distributed evenly in the shoot, as you can see, growing straight. Once the sunlight shines on the shoot at an angle, the IA molecules moves to the far side and induce elongation of the cells on that side. So when the sunlight deviates, the growth of the tip also deviates. So you can see here cell elongation. See, cell elongation results in bending of the shoot towards the sunlight. Further, it promotes root elongation in low concentration and shooting at higher concentration. It also hastens early rooting in propagation by cutting. So, auxins also help in promotion of root elongation and shooting at a higher concentration. So, that is providing propagation by cutting. Foliar spray of NAA and 2,4-D induces flowering in lychee and pineapple. So these two synthetic hormones also helps in flowering in certain plants like pineapple and lychee. It also prevents premature fruit drop in apples, pear and oranges, and also prevents formation of abscission layer. So it prevents the dropping of fruit or dropping of the leaves or premature planting by application of auxin. Auxin plays a role in elongation of cell. It is also known to increase the rate of respiration. Auxin breaks dormancy in seed and promote quick germination. Now this is again, you're familiar with breaking the dormancy. Certain seeds which undergo dormancy may not grow in the normal circumstances. So to break the dormancy period, auxin is applied, because of which the dormancy is also broken as well as it helps in quick germination. So here just added an animation image, okay, where you can see the quick elongation of the root taking place under the soil. Moving further, the second hormone, gibberellins. It is another growth promoting hormone and it is abundant in root tip and developing seeds. It shows non-polar transport through vascular tissue. Now, as we know, like auxin, gibberellin is also growth promoting hormone. It is abundantly found in the root tip and the developing seeds. Gibberellins was first isolated from fungus Gibberella fujikuroi by Japanese scientist Kurosawa in 1926. He observed that when rice plant was infected by fungus Gibberella fujikuroi, it shows extensive stem elongation called Beckane disease. So it was observed in the rice plant, the stem elongation seen, and the after the elongation stem breaks, which was termed later as Beckane disease. The crystalline form of gibberellins were isolated by Yabuta and Sumiki in 1938 from the fungus culture. They named it as gibberellin. It is synthesized in young leaves, seeds, roots, and stem tips. These are synthesized from mevalonic acid. More than 150 chemical types are known so far. GA3 is most common and biologically active empty form. Chemically, it contains a gibbon ring, a cyclic deuterpin with 4 isoprene units. So, to understand this term, I have added the image of gibberellin acid. So, you can see the structural formula of the gibberellic acid, and also another image showing the functions of gibberellin: cell elongation, seed germination, flower induction, breaking dormancy. Moving further, physiological effects and application of gibberellins. Dormancy of bud can be broken by gibberellin treatment. So, like auxin, even dormancy can be broken by gibberellin treatment. It can produce or promote seed germination in cereals like barley and wheat by synthesizing hydrolyzing enzyme amylase to produce sugar. Third, the most striking effect of it is the elongation of stem where internodes increase in length. So, if to increase the length of the stem or elongation of the stem, the gibberellin can be used where the internodes increase in size. It also promotes bolting, that is elongation of internode just prior to flowering in plants. Those with rosette habit, example beet, cabbage. It causes parthenocarpy in tomato, apple and pear and flowering in long day plants. Again, like auxins, even gibberellins promote parthenocarpy. It is used to increase the fruit size and bunch length of grapes. So it also helps to increase the size of the fruit. When gibberellins are applied on genetically dwarf plants like maize, the stem rapidly elongates and acquires the height of the normal tall trees or the varieties of maize. Now, as we know, the gibberellin helps in elongation of stem, so it is used in the dwarf plants. Application of gibberellin overcomes the requirement of vernalization, prevents abscission. Usually, it inhibits growth of root, delays senescence, and it also breaks dormancy of seed and hastens germination. Application of gibberellin causes production of male flowers on female plants. So even application of gibberellin helps in the production of male flowers on the female plant. Here I added an image showing the gibberellins which are used commercially in the spraying on Thompson's seedless grapes. Further, cytokinin, the third hormone. Again, like auxin and gibberellin, cytokinin is also a growth hormone. It is also another growth hormone that promotes cell division. Lethem coined the term cytokinin. The first cytokinin was discovered by Skoog and Miller in 1954 during investigation of nutritional requirement of callus tissue culture of Nicotiana tabacum, that is tobacco plant. They observed that callus proliferated when the nutrient medium was supplemented with coconut milk and degraded sample of DNA. They named it as kinetin. Chemically, kinins are 6-porphyryl aminopurine. First natural cytokinin was obtained from unripe maize grains by Lethem. It is known as zeatin. 6-benzyl adenine is a synthetic cytokinin hormone. 7 different types of cytokinins are recorded from the plant. So till now there are 7 different types of cytokinins, and 6-benzyl adenine, which is a synthetic cytokinin, that is artificially produced ones. Natural cytokinin are also reported from plants like banana flowers, apple and tomato fruits, coconut milk, etc. Here again I added the chemical structure of cytokinin, how chemically it appears, and another image showing the functions of cytokinin. nutrient response, apical dominance, vascular development, pathogen interaction. Organogenesis promotes cell division, meristem function, light mediated and development. Moving further, physiological effect and application of cytokinin. Besides cell division, it also promotes cell enlargement. Now, cell division, like auxins, here it also helps in cell enlargement. High cytokinin promote shooting, that is, help to increase the length of the shoot. A low ratio of cytokinin to auxin induces root development, but a high ratio causes buds and shoot to develop. That is, if there is a ratio of auxin and cytokinin, then it helps to induce root development, that is cytokinin to auxin. Or if it is a higher ratio, then bud and shoot to develop. So accordingly, if the ratio is adjusted, we can increase the root length or the shoot length. Cytokinin and auxin ratio and the interaction controls morphogenic differentiation, that is external appearance of the plant, the shoot length, that is the length of the stem or the root. It promotes the growth of lateral buds and controls apical dominance by cell division. It delays senescence, that is aging, and abscission process in the plant organs. Formation of intervascular cambium and expansion of cells are other functions. It also breaks dormancy and promotes the germination of seeds. Now, like auxin, gibberellin, even cytokinin helps in the to break dormancy and promotes germination. Cytokinin reverses apical dominance effect. It induces RNA synthesis and formation of intervascular cambium. Here again I added a simple image showing the auxins and the cytokinins ratio when it is uh, changed or the interaction controls the growth of the shoot or the root part. So rhizogenes, that is root length can be increased or the shoot length. Now the fourth hormone, that is ethylene. It is the only gaseous growth regulator. Now they are growth regulator and in the gaseous form. Denny in 1924 reported ethylene is effective in fruit ripening. Gain in 1934 established that plants naturally synthesize ethylene. Crocker in 1930 proposed that ethylene is also plant hormone responsible for fruit ripening. So you can see various scientists giving various uh, functions of ethylene. One by Denny, that is effective in fruit ripening. Gain, plant which produce or synthesize ethylene naturally. Crocker, plant hormone responsible for fruit ripening. It is a simple gaseous hydrocarbon with essential role in fruit ripening. The most widely used compound as a source of ethylene is ethopone. It is synthesized in roots, shoot, apical meristem, ripening fruits, etc. So here you can see the structural image of ethylene. CH2 double bond CH2. Beneficial effects. Again a simple image showing the beneficial effects. Triggers ripening, promotes color development in fruits, stimulates ripening in climatic fruits, degreening, stimulus dehiscence, alters sex expression, flower induction, reduces lodging of cereals. Moving further, physiological effects and application of ethylene. It promotes ripening of fruits like banana, apples and mangoes. The most important application of ethylene. It stimulates initiation of lateral roots in plants and breaks dormancy of bud and seed. It accelerates the abscission activity in leaves, flowers, and fruits by formation of abscission layer. Ethylene inhibits the growth of lateral buds and causes apical dominance and retards flowering. It is associated with enhancement of processes of senescence of plant organs. It inhibits flowering in most of the plants except pineapple. It causes epinasty, that is drooping of leaf and flowers. It increases activity of chlorophyllase enzyme causing degreening effect in banana and citrus fruits. Now what is this degreening? The definition: degreening, it is a process of decomposition of green pigment in fruits, usually by applying ethylene. This method is called trickle degreening. Now you can see in the present day some of the fruit industries of fruits which is manufactured, to degreen it, the ethylene is used so that the fruits can be marketed very quickly and easily. Further, the last fifth hormone, abscisic acid. It is natural growth inhibiting hormone. Now the auxin, gibberellin, cytokinin, they are growth hormone, whereas ethylene, abscisic acid, they are growth inhibiting hormone. Kans and Endicott in 1961 and 65 observed that shedding of cotton balls was due to chemical substance abscission 1 and 2. Waring 1963 isolated a substance from the buds of acer that can induce bud dormancy and named it dormin. These two identical chemical substances were given the common name abscisic acid. It is synthesized in leaves, fruits, roots, seeds, etc. Chemically, it is a 15-carbon sequiterpenoid and is synthesized from mevalonic acid. So again here you can see the structure of abscisic acid. Here we can see the abscission taking place, that is ABA, biotic stress, dormancy, germination, abscission, senescence, fruit ripening, abiotic stress. So here stress signaling, growth and development. Further, physiological effects and application of ABA, that is abscisic acid. It promotes abscission of leaves and induces dormancy in plants. Again, the most important function in case of ABA, that is, we can say the opposite of the growth promoters, that is auxin, gibberellins, cytokinins. It helps to overcome dormancy, promotes growth, whereas in case of ABA it promotes abscission and induces dormancy. It controls the dormancy in buds and seeds by inhibiting growth process. It accelerates the senescence of leaves, flowers and fruits. It inhibits and delays cell division and cell elongation and suppresses cambium activity by inhibiting mitosis in vascular cambium. ABA could cause efflux of potassium ions from the guard cells and result in closure of stomata. So it is known as anti-transparent. That is, ABA is also known as anti-transparent because it prevents the transpiration process from taking place. It acts as stress hormone by inducing the plant to bear adverse environmental condition. So hence it is also called as stress hormone. It inhibits flowering in long day plants but stimulates flowering in short day plants. Further, bioassays for here auxin, Avena curvature test, Gibberellins, alpha amylase bioassay, cytokinin, chlorophyll retention test, ethylene, triple P test, ABA, inhibition of alpha amylase test. Use of bioassay to measure the pharmacological activity of new chemically undefined substance, to investigate the function of endogenous mediators, to measure drug toxicity and unwanted effects, to measure the concentration of drugs and other active substances in the blood and other body fluids. This is just an additional information added over here. Lastly, assignment based on today's part of the lecture, that is about the growth hormones auxin, gibberellin, cytokinin, ethylene and abscisic acid. The 1 mark question and the short questions. Thank you all.